everyone. Welcome to October's Evening with Genetics. My name is Gladys Pryor. I am the Community Engagement Association for Baylor College of Medicine. And I want to welcome you to this month's Evening with Genetics called Celebrando Hispanic Heritage. What is in our genes? I do want to introduce our panelists today. So why don't we start with Dr. Montalvo-Ortiz. She is a neuroscientist and an assistant professor at Yale School of Medicine. She obtained her bachelor's in biology at the University of Puerto Rico, Rio Piedras campus, and her Ph.D. in neuroscience at Northwestern University at Chicago. Her NIH and VA-funded lab utilizes computational approaches to study the genomics and epigenomics of neuropsychiatric disorders with a focus on trauma and substance use disorders. She is the recipient of multiple awards, including the VA Career Development Award, NARSAD Young Investigator Award, the Faculty for Excellence in Diversity Initiative Fellowship, and the Lawrence Family Young Investigator Award. She also serves as the co-founder and co-leader of the Latin American Genomic Consortium at the Psychiatry Genomics Consortium Substance Use Disorders Epigenetics Work Group. Dr. Paola Giusti-Rodriguez is an assistant professor in the Department of Psychiatry at the University of Florida College of Medicine. Dr. Giusti-Rodriguez grew up in San Juan, Puerto Rico, where she earned her bachelor's in biology at the University of Puerto Rico, Rio Piedras. She completed her Ph.D. in cell and developmental biology at Harvard University, where her doctorate research focused on studying the molecular basis of neurodegeneration. Dr. Giusti-Rodriguez carried out her postdoctoral research with Dr. Patrick Sullivan at the University of North Carolina at Chapel Hill, where she employed the multi-parental collaborative cross mouse population to better understand antipsychotic side effects. Her work lies at the intersection between neuroscience and functional genomics, and she aims to integrate the tools and techniques of the fields to shed light on the genetics of neuropsychiatric disorders. In 2016, she was awarded a K01 Mentored Research Scientist Development Award by the National Institute of Mental Health for her work focused on functional genomics in psychiatric disorders. Dr. Giusti-Rodriguez is a co-founder and co-leader of the Latin American Genomic Consortium, where she seeks to increase the representation of research participants from Latinx admixed ancestry in genetic studies on neuropsychiatric disorders. Dr. Claudia Soler-Alfonso received her medical degree from El Bosque University Colombian School of Medicine in Bogota, Colombia in 2004. She also completed dual residency training in pediatrics and medical genetics at the University of Texas Medical School in Houston. She subsequently trained in biochemical genetics and metabolic disorders at the Children's Hospital of Philadelphia. Dr. Soler-Alfonso also authored numerous research items in the field of biochemical genetics and is a fellow of the American Board of Medical Genetics. She is currently an assistant professor of human genetics at Baylor College of Medicine and a metabolic geneticist at Texas Children's Hospital in Houston, Texas. Aside from her duties in Houston, Dr. Soler works closely with patients with inborn errors of metabolism in Colombia, where she leads efforts to improve the care in patients with genetic diseases. Her research interests include glycogen storage disorders, newborn screening, amongst others. And Liz Misrick is a certified genetic counselor at Texas Children's Hospital and Baylor College of Medicine. She received her bachelor's degree in sociology and geography from Stephen F. Austin State University and a master's degree in genetics counseling from the University of North Carolina at Greensboro. She works as an inpatient genetic counselor at Texas Children's Hospital. Her research interests are inpatient genetics and monogenetic diabetes. Liz provides mentorship, supervision, and education to genetic counseling students, medical students, pediatric residents, genetics fellows, laboratory fellows, and other learners in the community. She actively collaborates with researchers who identify and describing new rare diseases. Liz serves on the Molecular and Human Genetic and Genetic Counseling Program Diversity and Inclusion Committees. So we have a very impressive panel. And with that, I would like to hand it over to Dr. Montalvo-Ortiz.
Hi, everyone. So it's a pleasure to be here and talk about the origin and identity of us Latin Americans. And I'm going to talk about historical facts all the way to genetic data. So who are the Latin Americans? We know that we are a total of 654 million in the worldwide population, representing 8.4%. And we're among the most mixed populations worldwide. And also in the US, we are the fastest growing minority with 65.3 million people, which represents almost 20% of the US population. Here we're showing the proportions of Latin Americans by states in the US, with the highest being California, followed by Texas, Florida, and New York. And among the Latin Americans, the most prevalent that we have are Mexicans, followed by Puerto Ricans, Salvadorians, Cubans, Dominicans. But this just depicts the high diversity of the different Latin American populations in the US, but also, one of the most prevalent populations worldwide. So at school in Puerto Rico, I was born and raised in Puerto Rico, and I remember the first thing that we were taught about our identity as Latinos and Latin Americans, and to celebrate also our ethnicity, we were taught about the mix of the different races and how Latin Americans were defined as a mixture of different races. And we were taught about different social classifications of the descendants of mixed ancestry. And at the end of this call, we're gonna see how far we've come to these now obsolete classifications that are really not accurate. And we're gonna revisit the way we think about ancestry, the way that we think about Latin Americans, and we're gonna back this up by genetic data. So what is the genetic makeup of the Americas? There are 4 factors that influence the current genetic makeup of the Americas. First, we have variable native population density at the arrival of Europeans. We have also the influence of the European immigration during the colonization periods. We have the involvement of the New World in the African slave trade. And we have the admixture of natives, Europeans, and Africans. Particularly Latin Americans, this genetic makeup is known to be mostly composed by these 3 ancestral groups: natives or indigenous, Europeans, and Africans. So tens of millions of natives we know lives in the America, across all the Americas. And here I'm showing the distributions where we can see where we have some regions in Latin America where we have more prevalence of Native American populations compared to others. But just as an overview of the prevalence and also the differences in populations of the whole, across all the Americas. So definitely there is a great diversity in terms of subsistence and social organization, ranging from densely populated urban center depending on intensive agriculture like Mesoamerica, the Mayas, Aztecs, Zapotec, and the Andes, to large, scarcely populated areas occupied by small hunter-gatherers community like the Patagonia in North America. So I'm here showing just the flow of the different ancestry groups of the native populations across all the America. And we can see that also in the genetic composition of Native Americans across here. I'm just showing Central and South America, and the diversity of the different ancestry of the different populations of Native Americans across South America and Central America, just to have a sense of, the heterogeneity, how different they are just within the different Native American populations, how different we are genetically. And this can be explained by the geography. So how population diversification patterns reflect the geographic distribution. And this, as I was explaining before, how this could be explained by isolated communities living in the Amazons or Patagonia versus communities that were more accessible and that were able to communicate with each other. Now, the European settlement of the Americas, set in motion by Christopher Columbus landing in the Bahamas in 1492, was initially dominated by immigrants from Spain and Portugal. The Iberian expansion was followed by other Europeans, particularly the British, and to a lesser extent, the French and the Dutch. Here on the left, we show the different routes of Columbus to the America when he arrived during the colonization periods. And it is estimated that 500,000 Spanish and similar number of Portuguese migrated to the Americas, predominantly men. I am showing here the number of people specifically from Spain, and just to have an idea, within Spain where the most people migrated to the Americas. And also where in Latin America they were established. During the colonial period, another 1 million British, French, and Dutch arrived in their colonies, and following the arrival of European and colonization, 90% of the Native American population perished. The introduction of enslaved Africans to the Americas was initiated by the Spanish and the Portuguese early in the colonial period and gained strong impetus with the collapse of the native population. Slave trade expanded to other European nations to exploit the lands taken from the natives, like labor-intensive tropical plantations, like the sugarcane in the Caribbean. In many places, until the number of Africans greatly exceeded that of Europeans. Here I'm showing the number of Africans across the Americas and the different migration patterns from Africa to the Americas, and just to have an idea of the flow of those embarkments and disembarkments from African populations. So slave trade had a lot to do with the varying African ancestry proportions in Latin America. On the left, we show the triangular trade routes between continents during the transatlantic slave trade. We showed the embarkments and enslaved people embarking out of Africa, which were around 12.5 million, to disembarking, so people that were actually successfully arrived to the Americas. There were a lot of people that unfortunately died during this travels. And that reached around 10.5 million of African people disembarking in the Americas. And this happened between the 16th and 19th century. From these 12.5 that were originally embarked out of Africa to the Americas, at least 8 million were with the British flag. On the right, we have the estimated proportions of African ancestry across America and Atlantic Africa. And as similar as we were seeing with the native population, we also have a great diversity of the different ancestral groups among the African population. And that could be also influenced, reflected across the Americas, which was greatly influenced by the dynamic of the slave trade. And I just gave an overview of the native population, the influence before the Columbia era, the influence of the colonization, the European colonization to the Americas, the influence of the slave trade during colonization of the Africans that were taken out from Africa and put in as slaves in the Americas to work on the land. And as a result, we have 3 different ancestry groups that were mixed, but this was mostly predominantly in the Americas, where the social structure permitted for the mix of the different ancestry. This is not predominantly seen in the Americas, where there were more restrictions in the way the social structures work based on different ancestry groups. And here I'm just depicting the diverse admixture in Latin Americans, where we have the different proportions of ancestry groups: Native American in blue, European in pink, and African in green. We can see North America, Central America, South America, and the Antilles. And we're showing here the genetically estimated ancestry from these different groups of countries and dependencies across the American continent. And we can observe the varying proportions of ancestry groups across Central America, South America, and Caribbean. Here I'm describing another study looking at the diverse admixture in Latin American populations, where we show down here the different admixture plots for African, European, Native American, but also people from Colombia, Mexico, and Peru, and Puerto Rico. And we can see that sort of mix of 3 races based on the different proportions that we're seeing here, or ancestry groups, I must say, but also the variability in terms of the proportions of each of ancestry groups among the different countries, Latin American countries. And here we show what is called a karyogram, where it's basically the chromosome painting of the different ancestry-specific haplotypes or group of genes based on the different ancestry groups. And we see that there is a great diversity in terms of where different chunks of our chromosome are coming from different ancestry groups. And this is based on an individual from a US-based Latin American cohort. But I have to say that there is more to our genetic diversity as Latin Americans. I'm going to talk about the genetic remnants of the Manila galleon, which is less known as potential contributors to our genetic diversity. For 3 centuries, dating back to the 1500s, trading ships across the Pacific Oceans, and this came once or twice a year, from Manila, which is the Philippines capital, to Acapulco in Mexico. Trading consisted on silver, later bought by Ming China, and a variety of goods sold in the Americas, mainly in Mexico, New Spain, and Peru. And one of them is known to be the Manila, el manto de Manila, which is very typical of a flamenco dancer that is actually coming from those trades that were bought from China. Enslaved people in Manila, mostly Indonesian and Indian, were transported to Acapulco to work on domestic and merchant work. This influenced the ancestral profile of Mexico, where not only you have Native American or indigenous ancestry and African and European, but also East Asian ancestry. Here we show a map of Mexico depicting the East Asian ancestry proportion by region and city, where we have the higher prevalence in Guerrero, and a local ancestry karyogram of an individual from Guerrero, where we have the highest proportion of East Asian ancestry. And again, we not only see fragments of the chromosomes coming from African, European, and Native American, but also a big proportion from East Asian. And I have to say that this is rarely captured in genetic studies, not only studies considering the three-way admixture that is well established in Latin America, but also is lacking that potential fourth puzzle piece of our genetic background. And in this case, coming from centuries ago in history, in the colonial times, which is the East Asian contribution to our genetic diversity. And I have to say that, we're more than, ancestry groups. I think moving forward, the field should rethink the way we think about ancestry and think about ancestry instead of like continental groups or ancestry groups as a continuum. And this is thinking about how in the modern era, the admixture is, becoming more and more frequent with globalization. And I know there are different groups working on different modeling or methods that can allow it to capture this in genetic studies, and we hope we can, pick up the pace on this and modernize the field of genetics and in this context. So that's the end of my talk. I hope you learned something and that it wasn't too worrying.
Thank you so much. And I think now we are moving on to the next panelist, Paola.
Hi, everyone. Good evening. I think this Janitza set the perfect stage for my talk, which is focused on the Latin American Genomics Consortium. And so there's a pressing need to engage in more diverse, in research that involves more diverse research participants and investigators. And we have in the US, at least, we have also the genetics workforce is quite diverse still. And we, as Janitza explained, we have limited data and our understanding of the specific genetic susceptibilities of diverse and admixed populations. And achieving this involves not only investigators, but the communities that we are part of. And so in 2019, we co-founded the Latin American Genomics Consortium, which seeks to expand psychiatric genetics research in Latin America by engaging in collaborations across the region. And so I'm showing here, there's a lack of diversity in psychiatric genomic studies. And so, on the left, I'm showing a graph where we're showing first here, can I point? Yeah, great. Here I'm showing the populations of the world and the amount, like the proportion of the total population of the world that the different ancestral groups and the people from the different regions in the world occupy. On the left, you can see the distribution of these populations across psychiatric genomic studies for depression, schizophrenia, post-traumatic stress disorder, bipolar disorder, et cetera. And you can see that despite the fact that Europeans as a whole form just a smaller percentage of the world population, they are overrepresented across psychiatric genetic studies, while other populations such as populations from Hispanic ancestry, African ancestry, et cetera, are severely underrepresented in these studies. And this is problematic because, as I'm showing on the right, there are consequences to applying this genetic data in terms of clinical use. And so here I'm showing this graph from the study by Alicia Marty and colleagues, where they show that if you use genetic data that is derived from exclusively from European populations and then try to apply this data to individuals from other populations, it is not transferable. So you can you lose specificity, you lose, and this can lead to like an exacerbation of existing health disparities. So that if you move from using European data to look at, in this case, we're looking at polygenic risk score. So what is if you look at your genetics as a whole, your chance of having a disorder based on existing genetic data, it's very precise for Europeans, but as you move away, you see that you lose this precision. And this lack of diversity is also reflected in terms of the investigators that are carrying out these studies and the samples that are under study. And so here I'm showing both, this graph shows like for the Psychiatric Genomics Consortium, which is one of the largest consortium studying psychiatric disorders across the world. where were where most of the investigators came from, and they're primarily in the U.S. and in Western Europe. And you can see that there's a lot of regions in the world where you don't have as much representation, although this is a little out of date. And on the bottom right, you can see like where are the samples that are on the study, where are they coming from least, less sample to more samples. And you can see that the majority of the samples are coming from, again, Western Europe, the US, et cetera. And so there's many regions of the world that still are not well represented in these studies. And there are many factors that are behind this lack of representation. One of the reasons is that they see this underrepresentation of investigators participating in these studies is that there's a lack of investment in science in research and development across Latin America. And so in the US, about 2.8% of the gross domestic product is spent on research and development. And here I'm showing data from the World Bank for the most recent data that was available. And you can see that this differs greatly across Latin America for those countries that had data available. And so if you have a disinvestment, right, in research and development, this is gonna impact how many, like scientists you're training, how many people go on and pursue graduate degrees and go into the workforce for science and research. And we know that, this is just, a lot of these numbers are before the pandemic. The pandemic had definitely an impact on the economies across the Americas. And so, this just exacerbating these problems. And there are additional barriers, right, in terms of research capacity. There's a lack of collaborative opportunities across the region, partly because of this lack of investment. Many of the countries lack the adequate genomics and computing infrastructure to really be able to have this at scale. There's a lack of, for studying at mixed populations, like Janitza talked about earlier, really there's a lack of adequate methods to account for this complex admixture across the region. So you can't really box everyone together, because the composition of the genomic composition of the region is so complex. You really need tools that are designed for this purpose and that can really look at the interplay of the genetics. And so in terms of what data is available for psychiatric genomic studies in Latin America, because of this, the fact that there's a lack of investment in research, that leads to the fact that there's less participation, less samples that have come out of the region. And so here I've done a summary of some of the bigger studies that have come out, or some of the studies that are genome-wide, focused across a number of psychiatric disorders that are the standard for genomic studies, which is genome-wide association studies. And the reality is that for many of these psychiatric disorders, there's still a really big need to have larger proportions of research investigators and participants participating in these studies so that we can gather enough samples to capture all this admixture across the region. And we lack data for a lot of psychiatric disorders that are representative of these populations. This is changing, but it's still, we're long ways from having proportional representation of the region in psychiatric genetic studies. Again, this is, and some of the I think the best examples of what we're aiming for, right, is in Brazil, there's this Brazilian high-risk cohort for mental conditions. And in Mexico, there's this Mexican genomic database for addiction research, which are they're looking at large number of participants. They are tracking them. They're collecting phenotypic data. They collect genetic data, et cetera. And the idea is that we need more of these studies that involve biobanks and involve large numbers of participants, where you have phenotypic information, you have information on people's medical records and their different conditions in order to be able to put together a strong data set, have enough statistical power, and be able to inform psychiatric genetic studies across many psychiatric disorders. And so, I think a lot gets done despite the fact that there's very significant barriers and financial limitations, right, in Latin America. And so there's definitely room for growth. And one of the major goals of, of our consortium is to contribute to expanding these efforts. And as I mentioned, Dr. Atkinson is also in our call, but one of the biggest barriers that we face is the fact that analyzing at-mix samples is so difficult. So thankfully, there are more and better methods that have been coming out over the last few years. One of them, Tractor, developed by Dr. Atkinson, which help us parse apart this like complex admixture and help us look, at the contributions from the different ancestries to understand psychiatric disorders. And the reason, one of the, Janitza talked about the fact that it's a very complex problem when we think about the genetics, and the reality is that this has impacts that are that affect every stage of the analysis of the data. And so when we talk about the ancestry, right, we talked about the populations, but it matters in terms of the population that we're studying, right? It matters in terms of like the languages and the culture, like what are sort of cultural adaptations that happen for the various assessments that subjects have to participate in. It matters in terms of the genetic technology that is used to assay samples from these participants, because again, many of the arrays that are used to generate genetic data are biased for use mostly with European samples and not admixed samples or samples from other ancestries. It matters in terms of the reference panel. So how are you comparing the data that you're generating? What are you using as your kind of litmus test if it's a variant that is associated with a disorder or isn't? Again, showing the diversity of the populations, the indigenous populations in Latin America, you can see that it's very diverse. So it's not a one-size-fits-all kind of thing. It matters in terms of the methods that you use to study the data and the statistical methods to analyze and model the data. And obviously, all of that will have an impact on the results when you go and generate your data from your genome-wide association studies or whole genome sequencing, exome studies. And similarly, if you do functional annotation, what are the tissues, cells that you use to generate functional data that gives you information on the parts of our genome that don't encode proteins, which is really important for interpreting genetic results for GWAS studies. All of that matters, like what are the origin of the data? What are the subjects from which this data came from? And in the end, all of that feeds into as well, like clinical applications for precision medicine. And so if you have faults at each of these steps or any of these steps, it will impact how applicable this data is in terms of clinical applications of psychiatric genetic findings and precision medicine in the future. And so in 2019, as we mentioned, we founded the Latin American Genomics Consortium. We went from 25 original members to more than 125 active members now. We are spread across 9 Latin American countries, Puerto Rico, and the United States. Here I'm showing the institutions where our members are located at. And great, I think one of the biggest things is that 70% of our members are from Latin America. And our goal is to advance psychiatric genomic studies in Latin Americans. And so we aim to increase sample size. We are interested in contributing to methods development, in developing collaborative opportunities for recruitment and genotyping, and providing education and training for our members. And our members are distributed across Latin America, from Mexico to Ecuador. We have a number of affiliations. We have many that are Latin Americans but are in the US, but also that are working and living in Latin America. And we have members that go from being students to being faculty. And we have faculty that are young, like middle, and like advanced, faculty that have been faculty for a long time. We have a really good gender balance in our composition. And our members have a diverse research interest across all psychiatric disorders and brain cognition conditions, as well as in terms of the genetics, genomics, and epigenetics of psychiatric disorders. We are highly invested in collaborations. And so here I'm showing some of our collaborators, both within the US and across Latin America. There's the only way to get something like this done is to get like people to participate and to engage, knowing that they are going to get something out of it as well. And so we are always looking for new collaborations that will advance the goals of the members and of the consortium as a whole. And so some of the things that we've done, particularly in the past 2 years, is that we have been able to provide funds and facilitate connecting existing funds mostly from federal U.S. federal agencies like the National Institutes of Health to genotype samples, so we can generate genetic data across a number of psychiatric disorders involved, including schizophrenia, bipolar disorder, and substance use disorder. We've been able to gain data access for over 100,000 samples within the US and Latin America, which will enable, putting together a large number of samples in order to do these genetic studies. And we're doing various types of analysis right now and are started to organize by different psychiatric disorders in order to better coordinate amongst ourselves and advance each psychiatric disorder and the sample collection.
Thank you so much. That was so important. Next, we're going to go with Dr. Soler-Alfonso.
Thank you. I think it's fascinating, all this work that Dr. Montalvo, Dr. Giusti are really putting together, and it's really the best way to get into an actual story of the things that we see on the clinical side. It's really interesting to see the efforts that are made at the academic level and how these efforts are truly impactful when we're seeing patients with rare genetic conditions, and even when we're seeing novel disorders like the story I'm going to tell you about. So I'm going to tell you the story of Tango 2, and it's just by coincidence that I decided to talk about Tango 2. That's nothing to do with the fact that we're talking about Latin America, and in fact, nothing to do with Argentinian tango. But I decided to show you two cases because it truly takes two to tango. As you saw in the previous slides, there is an immense amount of collaboration that really needs to happen for the knowledge to really evolve. So the first case was a baby that was born without any significant complications, with no problems during the pregnancy. And then in the first 2 years of life, this baby was brought up to medical attention, mostly because she was experiencing problems with walking, and she had abnormal movements in the lower limbs. She also was showing that her head circumference or her head size wasn't growing accordingly with the charts. And she also had very low muscle tone, and her neck was also tilted, or she had torticollis. She had an MRI of the brain done, and the brain MRI showed some atrophy, which you can see here in this photograph. And what that means is that the brain size is not as big as we would expect for her age. When she was close to 31 months, she had a chickenpox infection, and she got very ill. And she came to the emergency department and got admitted to the hospital because she was noted to have significant muscle breakdown. And we call that rhabdomyolysis. And we measured different markers to look at muscle breakdown, and something called a CPK level was extremely high. It was 99,000 when it's usually less than 200. The muscle breakdown usually makes us worried about the heart. And we, in fact, noticed that this patient has significant heart rhythms, as you can see here in the EKG. And however, thankfully, this patient recovered, and she was able to go home, although the clinical recovery was very slow. Later on, despite the fact that she had this very significant illness and no one really understood what happened, she continued to have neurological regression. She was losing more milestones. She was not able to sit without assistance, and was not able to hold her head anymore. Unfortunately, at 36 months, the patient developed a new episode of muscle breakdown. This time, there was also significant cardiac arrest and significant cardiac arrhythmias, and she passed away, unfortunately, despite of the efforts at this very young age. Thankfully, she received a full genetic evaluation while she was hospitalized, and genetic testing showed us that she had a problem with a gene called Tango 2. The second case is actually quite similar. This was a baby that was born also without any complications. This baby was a little bit less ill than the previous baby during the first 1.5 or 2 years of life. And it was only showing some speech delays and some learning difficulties. At a later time, at 32 months, the patient started showing similar episodes to the previous case, with significant muscle weakness, pain in the lower extremities. The patient was also having episodes of slurred speech or dysarthria. And then it was obvious that all these episodes will come usually when there was a time of that illness or an infection. So at 4 years of age, the patient had a really bad throat infection, pharyngitis, and it was noted that the CPK level was extremely high, just like the previous patient. So there was significant muscle breakdown and also severe arrhythmias. And similarly, at 5 years old, there was another episode associated to a gastroenteritis. And unfortunately, this patient was also able to be discharged from the hospital. She was able to walk independently, but after this episode, she developed feeding difficulties and, lost some of the milestones. So it was clear that these patients were showing problems when they were ill and that their hearts will be fine. But whenever they had these metabolic problems or this crisis that we started calling metabolic crisis, with muscle breakdown, they will have significant cardiac arrhythmias and there will be a significant risk of death. So this patient is currently close to 8 years old, and she also has problems with hearing loss and low thyroid problems. But we also know now that this patient was found to have a genetic problem with the same gene, with the gene TANGO2. So what is this disease? So we were quite curious, right? I mean, we had patients that were showing these sort of patterns in muscle breakdown and neurological regression, and they were at risk of dying. But we also realized that these patients, as I'm going to show you here in this video, had episodes of abnormal movements that many times will be ignored by physicians or practitioners because they didn't look like much. And most of the patients had significant studies for seizures and everything was negative. But as you can see here, they had these episodes of head tilting, and it's quite obvious that it's affecting gait and balance. So it's a very debilitating disease. So as we move forward to understand this condition, then more case reports started evolving. And in 2016, one of our own Baylor investigators, Dr. Simalalani and Dr. Signoro with Dr. Kremer in the same year actually published several cases in different publications. And all the patients were very alike to what we had seen in clinic. They had this episodic muscle breakdown associated to the arrhythmias. So it was clear that this was an actual medical condition that was new and it was running around uncharacterized. So we started studying more patients, and at this point, there's approximately 80 patients reported in the medical literature that are affected with TANGO2. So what is this gene? Once we start seeing new genes that are coming out of the research pipeline with all these studies that Dr. Montalvo, Dr. Giusti are also collaborating with, and they help us understanding the meanings of genes and what they actually look like when they don't work in people, we started wondering about the function of this gene. And tango, as I mentioned earlier, doesn't have anything to do with the music, but it stands for Transport and Golgi Organization too. So that's quite a long name, right? So what we know that this gene does, or what we actually don't know yet, but we were quite interested because we think that this gene is very important for the way we move things within the cell. For some of you that are not that familiar with how the cell looks like, this is a cartoon of an animal cell. And some parts of the cell need molecules or chemicals to be transported in between the different compartments of the cells. So we think that this gene is very important for transporting different chemicals and signaling in between the Golgi complex and the mitochondria. And the mitochondria is part of the cell that is very important for energy generation. So we think this gene that we don't know yet, and we're doing studies, we think is very important for cellular traffic and the reason why people get so sick when they have genetic problems on this particular gene. So is it inherited? Well, yes. When we started studying families, we started realizing that there was more than one child affected. And we also understood that if the parents were what we call carriers, then they will have a chance of having a child with this genetic condition. And being a carrier parent only means that you have a working copy and a non-working copy of the gene. For every gene, we're supposed to get 2 copies, one from mom and one from dad. And having one working copy of the gene with parents that are carriers protect them from having any medical problem because they have at least one copy that is doing the job. So anyone that is a carrier for this disease and have a child with a carrier for the same disease will have around 25% chance of having another child with TANGO2. So we started seeing this by studying families, collecting samples, reaching out to people on the phone, collecting family histories, and the collaboration with families is so important. So we can start seeing the patterns emerging that tells us how this thing works like. So next time we speak to another family, we can be more informative. And this was actually quite daunting and scary for us as researchers working in this disease, because we knew these patients will have these metabolic phases for the compensations. They will go home, they will be semi-okay at home, but they were always at risk of having another crisis and having cardiac arrest, and they were at risk of dying. So we actually learned of a few deaths as we continue studying the disease. So we really needed to do something else to try to diagnose these children earlier and try to prevent these problems. So through all of these, we put together a natural history study, and the main investigator is Dr. Seema Lalani, who was one of the main authors for the initial publications. And Dr. Christina Miyak is a very talented cardiologist here at Texas Children's. And Dr. Clinton and myself at the two geneticists working, also collecting data, tabulating data, cataloging data, studying symptoms. And what we want to see is what the patients are alike to, what patients may be different in between them, what is it that is making them unique, and all of this with the goal of potentially gaining insight into the disease and determining if future treatments are available, or if there is a treatment that we may already have in our pockets that we can actually use for patients with TANGO2. The TANGO2 Research Foundation is actually an organization, a patient-based organization. So families that they started finding each other in social media, and now they have this wonderful organization that is doing a lot of fundraising. So that is also helping us for support and funding. And they really advocates for us to get this information and gather more. So we didn't know how frequent this was, because we started seeing more reports in the literature. And you learn now with Dr. Montalvo and Dr. Giusti's work that there is an immense amount of data out there. And we do different calculations in people in order to know how genes are flowing in between the population. So we had different tools as geneticists to know if there might be healthy carriers out there. And here are 2 examples of some of the healthy people databases. The Exome Aggregation Consortium is one of them, and the Genome Aggregation Database, the Genome AD is another one. But the representation of people with different diverse backgrounds is very scarce. So they're not the most up-to-date ones, but they help us. But we use other mathematical calculations to know how frequent this disease may be. And surprisingly, one thing that we learned about TANGO2, with the help of calculations of the Broad Institute and also rare genome projects, is that the disease can be as prevalent as 1 in 1.2 million. But surprisingly, and we all open our eyes, we learned that the prevalence in the Latino population can be as much as 1.5 million as well. But the carrier frequency is 1 or 600. Close to 1 in 614. So that means that if we had 600 people in this room and we were all of Latino origin, at least one of us would be a carrier. So let alone if we met someone else in this meeting and we had 1,200 people, so we have a chance of at least having 2 carriers in the same room, right? And potentially, if they get along, they may end up having a child together. So that gives you an idea of how frequent this may be. And it was actually, for us, a red flag, because that meant that there was probably more people and patients out there that were undiagnosed. So most of our data, when we did these math calculations and used similar tools to what Dr. Montalvo and Dr. Giusti were describing here, was enriched by people of Northern Mexican descent, mostly obviously because we practice in Houston, Texas. So most of our patients were from this area. So we looked in Mexico and we ran some numbers and we say, okay, so if there's at least one and almost 1.2 million people that had this disease in the Latino population, and in Mexico, there are close to 128 people, then at least there has to be at least 127 cases of Tango 2 in Mexico, right? And these are all very broad numbers that we calculated. And that also means that based on the Mexico's birth rate, there has to be at least 1 or 2 new cases per year. So this is actually quite scary for us because we know there are children out there in Mexico that may be passing away with this condition that we are not aware of and that we're not going to be able to find. We also don't have significant databases to explore and understanding what areas in Mexico this may be more prevalent or there may be more carriers. So as far as planning strategies for public health or other outreach opportunities, we are quite limited. And we try doing that. We have different databases from different people and at the Baylor level, at least. This is just one example of how underrepresented we are as Latinos. This is data from the Brittle Bone Disorders Consortium, which is a similar consortium to what Dr. Giusti was showing you for psychiatry disorders. And we see that the Hispanic, Latino, or Spanish origin individuals are extremely underrepresented. So we definitely need more representation. At this point, we have more than 100 families that are working with us in the natural history study, the registry of this disease. These are some of the patients that we met and some of the family conferences that have happened already, sponsored by the Tango 2 Research Foundation. But it's very important that we get more engagement of our Latino community, specifically even healthy individuals coming over and participating on us so we can continue learning the architecture of our genes, what makes us different, what we are alike to, so we can enhance all these research tools and approaches, and we can really impact our community and impact our public health approaches and policies and really save lives. I think at the end of the day, that's what truly matters. So I'm going to pass the ball to Liz, who is going to tell us a little bit more about resources and how to get engaged with research activities. And I hope that at the end you'll have at least an next step. So you are more interested, you can certainly contact us and know where to go. Thank you.
Thank you so much, Claudia. So I wanted to go over just some general resources to start getting engaged. And so one is the Latin American Professional Society of Genetic Counseling. So they do have a wonderful website that's available in English and in Spanish that has a bunch of resources, specifically this genetic counseling patient resources. And this is the link here. I'm hoping we'll be able to share our slides once the recording's available, so you guys will all have access to these resources going forward. The other one is the Mountain States Regional Genetics Network. So they have videos in Spanish that will walk you through what to expect at a typical genetics appointment, how we use medical interpreters, what newborn screening is, and has a general video about what genes, what chromosomes, what genetics is that can be really helpful to kind of give you the basic foundation for what genetics is, what we're talking about, and how to be familiar and comfortable with it in an upcoming appointment. And so to find a genetics clinic, there are many different ways. So you can go to the National Society of Genetic Counselors website. So all of these slides, I have the QR code where you are able to scan that code and be able to go directly to the website. So you're able to find these resources. And then there's also the link too. The ACMG, or American College of Medical Genetics and Genomics website, also has a list of all of the available sites that have genetics clinics as well. And in Texas specifically, the Mountain States Regional Genetics Network has a list of all the Texas locations genetics clinics, as well as the Texas Department of State Health Services. They also have a website that breaks it down by region, so you're able to find the closest genetics clinic in your region. And so how do you get involved? One of the biggest things that we've been trying to, I think in the country, is the All of Us research program. So anyone can join this program, healthy individuals, individuals with any medical conditions or genetic conditions. The goal here is to increase that diversity. So increase more of the underrepresented populations into our genetics databases, so that when we do get a diagnosis or we do get a genetic change, we're able to then better understand that information now that we have more information about that specific population. And so how it works is participants will share data, and this can be through just health surveys in general. So sometimes DNA is not needed, it's just doing health surveys as well. But also some will donate DNA samples as well. All of that data is protected, so it will not and is not shared. It's all under the guise of the password protected, all of that. And then the researchers will look at that data and study it, seeing if they see any patterns and be able to use that to help interpret any genetic findings we find. And if there isn't any information, the participants will get that information to them. And that will hopefully, our goal here is to help, like specifically, as we saw with Claudia's example of TANGO2, be able to find new genes and new diagnoses that can help explain what's going on for families. And so this is the sign up. So the sign up is in Spanish as well as in English. And so this is the QR code as well and the website. And so this is a good way to help get engaged and help with the diversity. So that's the biggest way to become involved. And I think like Paola had talked about with the Latin American Genetics Consortium, consortiums like that providing funds and manpower will help kind of create a more diversified genetics population that we have information or data to learn from. And so the goal of this talk is to potentially be a yearly talk that we give throughout Hispanic Heritage Month every year. And next year, we would like to have the talk in Spanish and then translated into English. So we wanted to ask the audience if there's any topics that you would like to learn about. That's the form to go to and also the QR code. We'll also be sending out a survey later to kind of ask for feedback. And then that's another place where you can give us topic ideas, because we want to hear from the community on what topics they would like to hear for next year. And so with that, I'll kind of open it up. Does anyone have any questions?
Yeah, these are fantastic talks. I feel like a lot of really different perspectives, like kind of the historical, the genetic, and kind of the clinical. What do you think are kind of the sort of biggest advantages from including more diversity and specifically sort of working with kind of Latin American populations in your various settings?
I can start. I think for me, on the clinical side, I see cute babies in the hospital, and it's really helpful when we get a result. Sometimes we can get more of an uncertain result. And so if we have more information from that specific population or ancestry or ethnicity, that may help inform us and be able to help interpret what we're finding on clinical genetic testing to help move forward.
Likewise, I will add that there is knowledge that evolves through time, that certain genetic changes that we know are disease-causing or problematic that may be restricted to certain ethnic groups or certain backgrounds, sometimes inform us about a specific treatment strategy. So sometimes it tells us that a patient may benefit from one treatment more than another based on what we know from their ethnic background. So sometimes we also learn that there are patients that process medications faster or slower. And this is true, particularly for medications in the psychiatry world. I know Dr. Gust is really liking what I'm saying. And then also medications that are important for blood clotting, for example. So if we start learning more about who we are as Latinos and we have more representation, it's going to be easier for researchers and clinicians to really translate that knowledge into a prescription or a treatment plan or a plan of care, and to really from there on develop more research to develop treatments for those things where we don't have still an idea what to do. That would be my added answer to that from Lisa's.
Makes sense. I guess, yeah, maybe following up on that, are there any challenges that you guys sort of see from sort of increased inclusion, and what would be, or sort of barriers? And do you have any ideas for kind of ways to overcome them?
I mentioned on the talk the money for a lot of the, at least for Latin America, I think like an underinvestment in science and like research and development. Obviously, that means that there's less money to do the type of genetic studies or clinical research needed to better understand, both rare disorders, psychiatric disorders, etc. And I think for genetic counseling, etc., there's not as many people trained in the region and with that speak multiple languages either. And so that impacts how not only the data that is being generated, but how that data is getting back to the patients. And so I think we need to make advances in those areas.
Yeah, I wanted to add to that events like this one that makes science more accessible to people that are either curious or that are in need to understanding better how, participating in genetics research can benefit them, like what they can take away from that. And I would say whether we like it or not, these clinical tools that are being developed will be implemented in the clinics. And in order to guarantee that these clinical tools will serve our population and that we will benefit from that, we need to participate and we need to be very well represented. And I will explain it why, just to highlight the urgency of this. So as Paola was saying, most of these data are being generated with mostly European populations. So what that means is that, for example, genes, or what we understand as genetic variants, may be more prevalent in European populations, but less or absent in populations like us. And when these genetic tools are developed, what they do is that they estimate a risk of having a disease or estimate the efficacy of a treatment. And if we're not very well represented there, what is going to happen is that it could be an overestimation or underestimation. It's a medication that could have higher side effects for us, that could make us sicker, or, the risk is not very well estimated and we might as well have the disease that we don't even know that, or not. So, that this can impact our daily lives. This can impact our health. And as some of us were saying, this can exacerbate already health disparities that, we already are on vulnerable positions on getting access to healthcare. So I strongly encourage people to participate in these genetic studies. It will help us to understand, who we are as Latin Americans, as my boring talk was saying, but also to generate those tools that, can improve our quality of lives and even better can help with whenever we're sick or our children are sick.
So we have 2 items in the Q&A panel. Lori says, I don't have a question, but I wanted to add that the Latin American Professional Society of Genetic Counseling website now allows you to search for Spanish-speaking and Portuguese-speaking genetics providers in the US and Latin America as well. So that's a really great resource. And then Marcia says, in clinic, what questions do you receive about genetic testing from Latino parents and families? Are there any common misconceptions about genetic testing in the community?
I can start because that reminded me of something I wanted to say. One thing that specifically working in Texas, I know some families worry about if genetic testing affects their immigration status or their ability to access resources. And immigration status does not affect any of our ability to see patients in clinic or order genetic testing. And so so that's something that is, I think, people can sometimes be uncomfortable with and not knowing whether or not they can seek services, but they absolutely can. And then also just, I think sometimes wondering like, how is that data used? And so when we do genetic testing, it's only used for that specific test and cannot be used for anything else.
And when we close that bridge and we are trying to understand something else that's going to be stated clearly to you. And I think it's very important that you use your voice. I think us as Latinos, we have lots of respect and venerance sometimes for medical services. We know for many of us, we grew up in areas where there was one doctor for a big area. So we have these different relationships with physicians. And I think we really need to be using our voices and really asking questions and asking more. Is this just for clinical care? Is this for research? And if you get the answer that this is for research, you really want to ask more. So what are you investigating? How are you investigating these? And don't be afraid. It's really our duty as doctors and researchers to tell you everything we're doing as much as possible in a way that you'll understand. And that be rest assured that we are tightly regulated by different agencies to be to use all the information and different samples that are collected to us for research in the ethical way, the most ethical way possible. And most of us also have a very high sense of ethics and how we handle research. And it's also important to be in a reputable place, a place that is strong doing research and work with doctors that will create those opportunities for asking questions back and forward. That's always a good sign, right? That you're in the right place. I will say too that sometimes we get confronted with the questions of feeling guilty whenever we realize that someone is a carrier for a genetic disease. And that is not only in our Latino community, it happens across the board. But I think it's important to know that genes change in all of us, and it's really no one's fault whenever we have a person that's a carrier. I personally am a carrier of 3 genetic diseases myself that I didn't know I was, right, until I got tested. So now I understand my risk of having children with these conditions. So it's important to have spaces where you feel safe and to use your voice and ask questions. Is probably the best advice that I can give to you to take in your pocket today.
Yeah, and there are at every major clinic, there are medical interpreters, and that's required by state and federal law, I believe. So language should not be a barrier either. I unfortunately don't speak Spanish, fluent Spanish, but I tell my families every time, like, leave me a voicemail in Spanish. I will find somebody to interpret it for me and call you back with an interpreter. So so that should be something that we will always have an interpreter available. And so always, like Claudia said, use your voice, ask questions. That's what we're here for.
Thank you. This was very exciting. And I feel very blessed to share this space with all of you and learn about your research and your passion for Latin American genetics. And I'm really thankful for the space.
No, the same. I feel honored to share with you, Claudia, that is like working directly with families, and I think that that's a more valuable job in my perspective. So, and thank you for inviting me to talk. Yeah.
All right. Well, thank you all for attending. We appreciate everyone who registered and came online to look at these very knowledgeable and amazing presentations that the panelists gave. So thank you to the panelists as well. I do want to remind everyone that this is a monthly series. So we will have one in November. That one is over I think it's cardiology. Yeah. So it's it will have to do with Marfan syndrome as well. So that is Tuesday, November 15th at 7 p.m. And I will put the link in the chat. Also, Liz, if you're able to pull up the From Stress to Strength program flyer, we'll also put that registration in the link. So this is to do with parents of teens and young adults with special needs. It is a two-part. So the first part is in October, I believe it's October 22nd, and then there's a second part in November. You don't have to register for both. It is one registration for both sessions, and you don't have to attend both. If you want to come to one or the other, that is perfectly fine. But we'll put those in the link. You will receive an email with all of the resources that we linked in the chat, also with the survey and the flyer for the From Stress to Strength program. With that, I want to thank everyone for attending again. We really appreciate you coming, and you enjoy the rest of your evening.